think I've heard of some negativity or anxiety about using Ad5 today and also T-cell responses, and I'm going to tell you about both of those today, so hopefully I can dispel some of this negativity or anxiety. So the title of my talk today is a non-replicating Ad5 vaccine for the treatment of HSV2 infection, but I'll also be touching on the protection in influenza as well. Okay, so Vaxart has developed a platform technology for the delivery of oral, prophylactic, and therapeutic vaccines. We believe, we administer the vaccine by tablet, and we believe the platform is suitable for delivery of many viral antigens. We are a clinical stage company. We've completed 2 H1N1 seasonal flu tablet vaccine trials, and the safety and immunogenicity profile was competitive with commercial vaccines after a single administration. We also have an advanced preclinical pipeline for seasonal influenza flu B, for norovirus and RSV, and we also have our first therapeutic candidate, HSV2 vaccine, in preclinical development. So just to give you some background on what I'll talk about today, first I'll give you some background on the technology. The vaccine is a non-replicating Ad5 TLR3 agonist platform. Then I'll go on and share with you some clinical data from our H1N1 tablet vaccine trials. And then at the end, I'll switch tracks and talk to you about our preclinical work characterizing potential T-cell antigens for inclusion in our therapeutic HSV2 vaccine. So some background first. So the vaccine is delivered by enteric-coated tablet, and that's to protect the virus from the acidic environment of the stomach. Once the tablet has arrived in the small intestine, it dissolves, and then the virus is then allowed to deliver the adjuvant and antigen to the mucosal epithelium for expression. And it's the double-stranded RNA adjuvant, the TLR3 agonist, that begins the immune cascade, and so we get the development of strong T and B cell responses. So one of the big advantages, of course, of a tablet vaccine is that it's needle-free. This leads to patient acceptance and greater patient compliance, we believe. It also leads to an ease of administration. There's no medical personnel involved. The tablet can be mailed, if need be, in a pandemic situation. And there's no needle stick biohazards, nor is there any difficulty in disposal of sharps. Vaxart's tablet vaccine is incredibly stable. We're now stable at room temperature for greater than 1 year. And room temperature stability leads to no cold chain. No cold chain means we've got an ease of distribution. It's a lot easier to transport a non-refrigerated product, making logistics simpler, and of course, costs are reduced. So to move on to our clinical data, we have completed 2 H1N1 phase 1 trials. They have been randomized, double-blind, and placebo-controlled trials. In the first trial, we had groups of either 1E9 or 1E10 IU doses, along with placebo. And in the second trial, we had a group of 1E11 IU as well as placebo. And the purpose of these trials was to determine safety and immunogenicity and to determine the optimal dose range for the vaccine. So our vaccine is incredibly stable, or it has an excellent safety profile. All the AEs were mild except for 1 moderate headache and 1 moderate abdominal pain. And the number of AEs between the treated vaccine groups and the placebo groups were not significantly different. Moving on to the immunogenicity data, the correlative protection in flu is a 4-fold rise in neutralizing antibody titers. We saw a 75% of subjects seroconverted by HAI after a single dose, and 92% of subjects seroconverted after using a microneutralization assay. So as well as being able to produce systemic neutralizing responses, IgG responses, we're also able to induce a strong mucosal IgA response. And this is shown here in this graph. These are antibody secreting cells for IgA, so we're looking at pre-plasmablasts. And you can see a nice dose-dependent response here. And then in contrast, when 4 volunteers were given the IM vaccine, or the injectable vaccine, they did not induce a mucosal response. So we can produce systemic neutralizing antibody responses, we can induce mucosal IgA responses, but what about cell-mediated T-cell responses? The answer is yes, we can induce some nice T-cell responses. Here's a dose-dependent response here. This is shown by interferon gamma ELISpot results. But not only can we induce T-cells, these T-cells are also mucosal homing. And we know that because they express the receptor alpha-4 beta-7. So what you're looking at here is a FACS plot of interferon gamma producing CD8 T-cells on day 0 before vaccination and day 7 after vaccination. And then again, you can see a nice increase in alpha-4 beta-7 expression. So that got us thinking into what other diseases could benefit from mucosal homing T-cells. Clearly, flu and T-cells are not particularly important, although people are changing their minds now as regards cross-reactivity in flu vaccines. But we knew from work from Connie Trimble and others that alpha-4 beta-7 is required by T-cells to enter the genital tract. And then more recent work by Shannon et al. Has shown that PBMCs from asymptomatic HSV2 subjects are actually also expressing alpha-4 beta-7. So taking all this together, Vaxart's therapeutic HSV2 vaccine program was born. So I'll change tracks here and talk to you about characterizing potential T-cell antigens for inclusion in our therapeutic HSV2 vaccine. So before I do that, though, I'd just like to review the pathogenesis of HSV. So when the virus enters the genital tract, it'll go ahead and infect the epithelial cells. There it replicates and then lyses the cells, causing lesions. The virus is then allowed to escape and enters the sensory neurons. And it's there that it sets up latency. After some sort of environmental stress, the virus can reactivate, come out of the neuron, and again go on and infect the epithelium, again causing lysis of the cells and lesions. And this is the common recurrences that you see in HSV2 infection. So with a therapeutic vaccine, what we hope to do is we hope to induce cytotoxic T cells that can keep the virus in check or keep the virus in latency at the level of the neuron. We also want cytotoxic T cells to be able to kill the infected epithelial cells, but we also would like some antibodies too, some neutralizing antibodies, because these can help neutralize the virus when it pops out of latency from the neuron here before it enters or infects the epithelial cell. But we don't just want cytotoxic CD8s, we also want these cells to be polyfunctional. And what do I mean by polyfunctional? Polyfunctional cells are cells that express 3 or more cytokines or functions. In our case, we're looking for IL-2, TNF-alpha, interferon gamma, and the cytotoxic marker CD107A. Why do we believe we need polyfunctional cells? That's because they've been correlated with efficacious vaccines such as yellow fever and smallpox, and also HIV non-progressors also have an increase in these polyfunctional cells. As well, asymptomatic HSV2 seropositive subjects, that's subjects that are controlling HSV, also have an increase in these polyfunctional CD8 T cells. So Vaxart's vaccine contains GD and ICP0. GD is a late gene. It's an envelope glycoprotein, which makes it a good neutralizing antibody target. And GD is used to enter host cells. And it also has documented T cell epitopes in humans. ICP0, on the other hand, is an immediate early gene. So it's the first gene expressed, or at least one of the first genes expressed as HSV reactivates. And because of that, we believe we'll get a rapid immune response to the antigen, as it's going to be presented early on these infected cells. And that's an important point. It also has documented CD8 T cell epitopes in humans. So we wanted to go on and test the efficacy of our vaccine, and we did this in collaboration with Rhonda Cardin and David Bernstein at Children's Hospital Medical Center in Cincinnati. And what they used is a therapeutic guinea pig model to test different vaccines of interest. So the model is we infect with HSV2. By around day 7, the disease peaks, and then by day 14, the disease has resolved. At day 14, 21, and 28 is when we vaccinate. And then from 28 to 63 days, we monitor lesion scores and shedding of the virus. So this is just the results of that experiment. You're looking at cumulative lesion scores on the y-axis, days post-infection on the x. And it doesn't seem to matter whether we deliver the virus or the vaccine orally or intravaginally. We see about a 35% decrease in scores compared to the guinea pigs that were not given any vaccine. And so we were a little disappointed with this. 35% wasn't what we were hoping for. Other therapeutic vaccines that have gone to the clinic have seen around a 50% decrease in guinea pig lesion scores. And so we wanted to try and understand the quality of the T cells that were being induced by GD and ICP0. And that's, we were wondering whether we were in fact inducing these CD8 polyfunctional cells, which is what we were hoping for. And to do that, we used a mouse immunogenicity model. And this, we vaccinated the mice at day 0, day 7, and day 14, and then waited another 2 weeks till day 28. Then we isolated the genital tract and isolated the T cells from the genital tract of these mice. And then we carried out multicolor flow cytometry, staining for CD4, CD8, interferon, TNF, IL-2, and CD107A, which is the cytotoxic marker. So we're trying to find polyfunctional T cells. We didn't just use normal inbred mouse strains because we didn't want to bias our response here. Using just a BALB/c mice, we've just got one haplotype, one MHC haplotype. And so we wanted to be able to mimic the immune response of the outbred human population. And so we went ahead and used these recently available Jackson diversity outbred mice. These mice are generated by crossing these 8 inbred strains to create a collaborative cross. And then these collaborative cross mice are just randomly bred to produce the diversity outbred mice, or the JDO mice. So this is the results of the multicolor flow cytometry. It looks a bit scary, so I'll talk you through it. So this is the analysis of the T cells that are trafficking back to the genital tract after immunization with GD alone. You're looking at percent CD4 T cells, or percent CD8 T cells producing cytokines here. And on the x-axis, we're looking at different cytokine populations. So for instance, in the yellow here, all of these cells are producing each one of these 4 functions or cytokines. So the yellow and blue are the polyfunctional cells. That's what we want to look for, that's what we want to see. The orange are the bifunctional cells, and the tan is the monofunctional cells. And each one of these gray circles is an individual mouse that we've isolated T cells from. So I think it's clear to see that we've got a lot more CD8 cytokine-producing T cells after vaccination with GD. That's reflected down here in these pie charts. There's about 21.7% of CD8 T cells producing cytokines compared to only 4.63% of CD4 T cells producing cytokines. And what was also nice to see is that we're getting a nice population here and here of polyfunctional cells with the CD8s. and that's really not reflected in the CD4s. So GD looks like a pretty decent antigen. And it's actually the antigen that has been included in recent trials in the prophylactic HSV2 vaccines, which didn't work. Just to bear that in mind. We went on and looked at ICP0 in the same sort of assay. So these are mice that are just vaccinated with ICP0. Again, we're looking at T cells that have trafficked into the genital tract. And I think what's also clear here is that we have an equivalent response between CD4s and CD8s. And that response is not very high. We've got about 8.6% of the CD4 cells producing cytokines that have trafficked to the genital tract, and about 7.6% of CD8s. And that's in comparison to the 21.7 that we saw with GD. Not only that, but the cells do not appear to be polyfunctional, either for CD4 or CD8. The majority of the T cells seem to be producing either, or seem to be bifunctional or monofunctional, and that's the same for CD4 and CD8. And that's reflected in these pie charts. So here we're looking at the polyfunctional cells. These are the bifunctional and the monofunctional. So you can see for both CD4 and CD8, the majority of the cells are either monofunctional or bifunctional. Not what we want. So this perhaps explains our disappointing efficacy results. So we also looked at the total amount of interferon gamma that was being produced after vaccination, and we did this by looking or restimulating the splenocytes from these mice. And you can see, basically, I think it reflects what we've seen in the multicolor flow cytometry graphs, in that GD is a lot more immunogenic than ICP0 here. And bear in mind, these are outbred mice. So this is why you're seeing a real diverse amount of interferon gamma being produced. If these were an inbred strain, you'd expect it to look kind of all clustered down here, these spots. Interestingly, that's what's happening with ICP0. So ICP0 seems to be not very immunogenic across a lot of different MHCs. So we then went back to our screens and we pulled out another antigen, which we've called VX83859. And you can see here when we're looking at the total amount of interferon gamma produced, that this 3859 antigen appears to be really superior to GD and to ICP0. So it's producing a lot more of total amount of interferon, but is this reflected by CD4s or CD8s? So again, we went back to our multifunctional flow cytometry, and after vaccination with just 3859 alone, you can see that we have a lot more CD8 T cells producing cytokines compared to CD4. And this actually is a whopping amount. I mean, we've got around 65.7% of the CD8 T cells that are trafficking back to the genital tract producing cytokines. And that's in comparison to around the 20% we saw for GD and the 8% that we saw for ICP0. Not only that, but we're getting a really nice polyfunctional response too. And that response also appears to be immunodominant across a number of different MHC haplotypes. So we have the responses all clustered up here. We don't have very many down here. So these pie charts just depict the proportions of these different populations. So you can see the blue and the yellow is what we're looking for. These are the polyfunctional cells. So not only do we have 65.7% of cells producing cytokines, around 50% of them are actually polyfunctional. So just to summarize those results, if we vaccinate with GD alone, we get around 21% of CD8 cells producing cytokines, and of those, around 33% are polyfunctional. However, if we vaccinate with ICP0, we only get around 7.6% that are CD8 cells, and only a quarter of those are polyfunctional in their cytokine production. However, when we vaccinate with 3859, we're seeing around 65.7% of cells, or CD8 cells, producing cytokines, and literally half of those are polyfunctional. So we're eagerly awaiting the efficacy results of GD plus 3859, and we should get those next month, actually. So just to summarize, our clinical trial results using our tablet influenza vaccine, we were able to produce neutralizing antibody responses that were comparable to seasonal IM vaccines. We were also able to elicit mucosal IgA responses. And when we looked at T-cell responses, we saw CD8 producing interferon gamma cells that were also mucosal homing. And that led us to our preclinical program for the HSV2 therapeutic vaccine, where T-cells, especially CD8 T-cells, are very important. And we have identified a potential T-cell antigen with immunodominant epitopes. It elicits a high percentage of polyfunctional CD8 cytotoxic T-cells. And the efficacy study, as I just mentioned, is ongoing with GD plus 3859. So just want to thank our collaborators, David Bernstein and Rhonda Cardin at Children's Hospital Medical Center in Cincinnati. And I'll leave it there. Thank you.
Thank you very much.
Why don't you use an enteric adenome? Why use an Ad5?
Why don't I use what, sorry?
An enteric adenovirus.
Yeah, so that's a good question. So historically, that's not how the technology started out, but that would make a lot more sense, right?
Yeah.
So we are investigating that too.
Yeah, because Walter Reed have two enteric adeno vaccines they use.
Right. I think they're replicating.
Yeah.
Yeah. So replicating obviously is a problem because
But they use as live vaccines in troops.
Right. But you get a lot of anti-ad responses, right? Oh, you mean because it's an ad vaccine?
Yeah, I mean it's a live vaccine.
Yeah. No, they definitely do that, and yeah, it's good. But what we're trying to do is we're trying to create a platform.
Yeah.
That we can give multiple antigens and multiple vaccines. Right. Once you've had that ad vaccine, we can't give something like
Extra. I don't think anyone's ever tried it. I mean, those Walter Reed vaccines are very good, but no one's ever tried to make them as recombinant vaccines to put other antigens on top of them. At least to my knowledge.
So there's lots of ad vaccines that are given IM, and you get tremendous neutralizing antibody responses to the ad itself. So our idea is to give it orally, and you just don't.
I agree. Yes.
You don't see it as much.
Any other questions? Yes.
That's very nice tablet vaccine. I think so. That's actually indicated you do mucosal inhalation.
Right.
This is oral.
Right.
It's one single tablet.
Right.
So you have a specific techniques formulating oral tablet to try.
Right. So the technology is really in the enteric coating. So it needs to be able to open up in the right part of the small intestine. So it's it's pH dependent, and we're hoping to open up in the ileum, and we have seen we have tracked the tablets and have seen that happen. That's because that's where all of those patches and immune sites are, or most of them.
Yes.
Was there any IgA production when you use the GP protein or other?
So I haven't looked at IgA in the HSV system, but it'd be interesting to look at.
So is that depends most of the antigen under the delivery system to get the IgA or different kind of response. The antigen is play a main role.
It may, we've looked at IgA in the flu vaccine and in our preclinical norovirus, two different antigens, and we get similar IgA responses. But I'm sure, it could be antigen dependent. I mean, yeah, it could definitely be antigen dependent.
You have the same delivery system between the influenza and the herpes. You use the same system.
Exactly the same platform. We just slot in a different antigen of choice, and that's really where we think, that's where the technology has huge advantages.
Yes, so you don't have IgA because of the platform, like because of the antigens.
We do often,
get the IgA responses with the influenza,
Yes, exactly. So I did show some IgA responses there with the, in the
but
humans.
not with the GP or other.
I haven't looked for GP,
very
but I have, we have looked with norovirus and we get nice IgA responses, which is obviously what we really want with norovirus. It's a gut pathogen. We want dimeric IgA, and we're definitely eliciting that.
Okay, so it's still the platform.
Yeah. I think it's the mucosal delivery,
Okay.
more than the antigen itself.
Any other question? Yes.
The mice before immunization were exposed to HSV or the
No, it's pure immunogenicity, because I wouldn't be